Good afternoon. Yeah, you can all hear me okay? Thank you all for your attention. As Victor mentioned, I've worked in the industry for over 30 years. So today, I hope you'll indulge me a little. It's going to be part history lesson, maybe a reflection on our industry and how we work today. And then I'll go into a little bit of formulation development. I'll probably skip through a few slides just with time to try and keep us on track. Wrong way. Okay, Age of Big Pharma. I'd like to start with two companies, SmithKline and French and Beecham's. SmithKline and French being an American company, famous for Tagamet. They had a development facility in the United Kingdom, and Beecham's that was famous for antibiotics. And the two companies merged to form the company SmithKline Beecham. And that's where I started my career. And we had 5 development campuses in the UK, all dotted around London. So 5, each with maybe 200 to 300 staff. This is back in the time when big pharma companies were totally self-sufficient. So we had pharmaceutical development, chemical development, DMPK, toxicology. We had our own animal houses. We had clinical development. We even had some beds for volunteer studies, regulatory affairs, discovery, pharmacology, everyone. We did not have to outsource at all. And it was a different time. When I had a challenge, I can ring up a colleague in the company, maybe in chemical development, and ask them, how is the drug synthesis progressing? Do you have the results on the yield? Is your degradation profile the same as before? Have you measured the particle size yet? And we're all going in the same direction. We worked for the same company, and all of our interactions were purely on a technical level. Another example might be with tox. We just heard a very nice presentation on tox coverage, which is very important for my staff. I needed to know that my people were safe and that we were using the right PPE for the studies. So I could ring my colleagues and say, how is the tox study progressing? Are we able to start manufacturing this? Apologies for this being a little bit UK-centric, but here's a map of the UK. Some of the companies you may recognize, some you may not. But if I take a few examples, let's take Glaxo. So Glaxo bought Allen and Hambrys. They merged with Burroughs Wellcome to form Glaxo Wellcome. They then merged with us to form GlaxoSmithKline. And that was a campus that was probably about 4,000 people in the UK. A few others, you'll probably have heard of Pfizer. Pfizer bought Wyeth. They bought Pharmacia. They had the largest pharmaceutical campus in the UK back in the day. Nowadays, they don't even exist in the UK. Last one, I'll just say, maybe the Swedish company Astra, merging with the British company Zeneca to form AstraZeneca. So this was a time when there was a lot of money and all of the companies were self-sufficient. I'm going to jump forwards. This is what we look like today, and you may recognize some of these, you may not. This is a company that I used to work for, Almac. But we have Patheon, we have Catalent. These are global CDMO companies now. And if we look at what's left of the big pharma industry in the UK, these are the only 3 companies that still exist there. Bristol Myers Squibb has a small area up in Morton, AstraZeneca and Glaxo. It's a very, very different work environment for the formulation scientists of today. And there's some advantages and there's some disadvantages with that. CDMO, Contract Development and Manufacturing Organization. So combined development and commercial operations. This has resulted in a reduction in the number of sites and equipment in some of our facilities. And quite often they'll push for single quality systems so that the commercial people, they want to sort of drive things because they're the ones that make all of the money. The scale of operation has changed as well. It's probably got smaller, which is a good thing. Because what we find is our customers come along with the smallest amount of API possible. And they ask us to perform miracles with this. You know, can you manufacture all these supplies? So we've seen the equipment that has been brought in get smaller, and we're just making batches that are fit for purpose. We aren't thinking about blockbusters at this point of time within the CDMO industry. We've also seen a change in indications. There's a lot more orphan indications, which are for very small populations in society. So we don't need these huge batches anymore. I can give you an example of one of the commercial products that we manufactured. We only made 3 batches a year, and each batch was only 5 kilograms in size, and those were just capsules. That's how small the population was for the indication that was required. And then there's also a push for pediatric medicines. You will not be able to get your adult indication filed with the regulatory agencies unless you have a PIP, Pediatric Investigation Plan. And again, the market volumes for these are very, very small. So we're starting to see a lot of the manufacture in drug product getting smaller and smaller. Then we've got the complexity of the operation. And it was interesting to hear the earlier talk on HBELs. There have been times when I have pushed customers that please, 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 will you go and see this nice lady here and get a report and give her your tox information, because I need a report to cover all of my staff. And it has been a bit of a push at times. Back in the day of big pharma, the corporate ethos was safety. So I knew that my tox department would have to generate that information, that they would have to give me that report that would cover all of the PPE for my staff. Now it's changed. Now I am pushing my customers to say, please, will you generate this information? Because they will try to take shortcuts. They will try to produce as little API as possible because it's expensive. And what they will do is they will default to say, oh, can you not run contained manufacturing where you protect the operator from everything? It's possible, but it becomes very expensive. Now we're talking about scientists having to work in full air suits with HEPA filters, contained equipment and valves, which makes the manufacture a lot more difficult and complicated, especially when you're trying to develop a process for the first time. And then lastly, staff. And I've seen this as we've moved to CDMOs where some of the companies, they don't have some of the staff members in the right functions that they need. So they may say to somebody, well, I know you're an analytical chemist, but can you go and become a formulation scientist, please? And we see this all the time in the training programs, people turning up, and they've been asked to move into a different area. And they just sometimes don't have the right background education. You can still run a 3-day course, but you'll not teach them about pharmaceutics in that period of time. So, my second history lesson is just about the products that we manufacture. And every year, the FDA produce a report on the number of new drug applications that are approved. And they have archives that run back to 2004. And these numbers are actually quite small. If you go back to the 70s and 80s and 90s, we were probably much more prolific as an industry, but we are actually approving less. And part of that is because of the complexity of the filings and the amount of information that is required to get a drug through the regulatory system. But I'm going to focus on just the final year, 2013. And not a particularly good year for the FDA. They approved 27 new molecular entities. And a few interesting facts about that. Of those 27, 17 were tablets and capsules, 7 were injectables, a couple of inhaled and topical. So at this point in time, solid oral dosage forms are still the dominant formulation that we are working on. And injections are starting to come through. Interestingly, 33% of those were for rare diseases. And if you understand the definition of a rare disease, that's less than 200,000 population in the US. Out of 350 million, it's very, very small volumes, very small market volumes. So we're starting to see a lot of the medicines that are coming through, they're much more niche and for orphan indications. The other thing that's interesting is the regulatory pathways. So the FDA had a total of 4 different review pathways that would speed things up. Fast track, breakthrough therapy, priority review, and accelerated approval. And what is a little bit concerning is the fact that nearly half of the applications are on those expedited pathways. We're getting to a point now where the regulatory authorities are swamped. And if you don't get your molecule on one of those pathways, it can sit there in the review process for a very, very long time. I'm going to jump forwards by another 10 years from 2013 to 2022. The regulatory agencies have been under pressure to be as productive as possible and get as many molecules through the system as possible. So this is definitely an improvement over the last 10 years. And if we look carefully at 2022, we have 37 new molecules in 2023. The types of dosage form has changed. We've now got 12 oral dosage forms, but we've got 21 injectables. And the reason that number's like that is because we're now seeing more biopharm molecules coming through. All of the biological molecules are going to be injectables. They're not going to be solid oral dose forms. And that's why there's a big switch around in those numbers. The next thing is more than 50% were for drugs for rare diseases. So we're continuing on with that situation where we've got new medicines coming out, but they're all for very niche indications, and they're all very small volume, so it needs small-scale manufacture. I think the thing that's most concerning would be expedited development. So we've now got 65% of approvals going through the expedited review pathways. And if I pick up a question that came earlier from Bashir, you asked about the COVID vaccines and why can't we just do this fast? I can be fairly certain that when those injections went through the system, all of the other programs were probably put on halt. The agencies themselves were so swamped with these injections coming through, they did not have time to review all of the others. So that's one of the things going for the future. You know, if your molecule isn't for an unmet need, something very important about it, it will take a long time to get through the regulatory process. That is just the reality of it. So those are my history lessons finished. And despite the fact that injections were starting to win, I'm going to stick on tablets. The reason being, as Sudhakar mentioned at the very start, 90% of medicines on the market today are solid oral dose forms. And if you consider your parents or your grandparents and the situation at home, I know my mother-in-law counts out the tablets for my father-in-law at the breakfast table, his tablets and his capsules for his blood thinning and his hypertension. I'm sure we all have family who take exactly the same. Tablets aren't going to disappear. They are the most important dosage form. And not just because of convenience and patient compliance, but for technical reasons. Now, you'll have seen the bioclassification system presented a couple of times today, and I just want to remind everybody what happens when we are taking tablet orally. It has to be in solution, and it has to be permeable, and then it will cross through the gut into the blood system to be transferred around the body. There's one other important thing, and that is dissolution rate. And you'll start to see why some of the technologies come through to increase dissolution rate. The stomach mixes up the stomach contents along with our tablet, and we'll have peristaltic waves go up and down. We'll create a chime, and it slowly just gets outside of the pyloric sphincter, and it gets into the small intestine. There is no absorption from the stomach. And if I take you back to biology at school, if you remember the intestine, it's got lots of little microvilli in it, and they always used to tell you that if you took the small intestine and stretched it, it would be the size of the Camp Nou. It is the size of a football pitch, and therefore it's the perfect organ for absorption because we absorb polysaccharides, we absorb proteins, we absorb fats. We even have special organs to help with water-insoluble materials like fats because we've got the gallbladder which secretes bile acids. But all of the absorption takes place in the small intestine. And as we come out of the stomach, the first thing we hit is the duodenum, and then we get to the ileum and the jejunum, okay? And we're probably at about a pH of 4.5 to 5.5 to 6.5 as you go through the small intestine. That is where the absorption takes place. Now, you can go to certain companies who will actually work out where the absorption window is because it doesn't necessarily get absorbed the whole way down. You can do gamma scintigraphy studies, and it works off scanners. They take the medication that has got technetium-99 in it, and they can actually see where it gets absorbed in the small intestine. Now, the reason that is important is if the drug in the tablet is broken down, it disintegrates, and it still leaves the stomach as a solid, it will not get absorbed. It has to be in solution. It may be starting to dissolve, but if it doesn't dissolve fast enough and it passes by the absorption window in the small intestine, then it just goes round and round and up the large intestine and out the end. So there's something called bioavailability, and we can do studies to measure the bioavailability of a drug. You can do an injection, and then you give a solid oral dose form. And what you're looking for is what percentage of the drug is actually absorbed from the stomach and gets into the bloodstream. Really good example, something that's going to have 100% bioavailability. Doesn't happen as that often. But there are still drugs out in the market that have a bioavailability of only 1%. But what we're trying to do is we're trying to maximize that all the time. The more drug that we can get absorbed in the intestine, the smaller the dose, the lower the cost of goods. That's a good thing. So let's start with our formulation development on a simple tablet. First thing in our tablet, drug, obviously. And if you can imagine for a moment taking this drug and you were able to make it into a tablet, the first thing is you wouldn't be able to pick it up. You need a pair of tweezers or chopsticks, because if you only have a dose of 10 milligrams or 20 milligrams, the tablet's going to be so small you can hardly lift it up if you were able to make it into a tablet. So invariably the next thing that goes into a formulation is a diluent. And as soon as you bring those two things together, you can appreciate we have to mix this. We have to mix this uniformly, because if we don't, then when we make our batch of tablets or capsules, we're going to have some super potent dose forms, and we're going to have some subpotent dose forms. As soon as you bring two materials together and mix them, the next thing to think about is, yes, but they can segregate as well, and they can demix. So you probably need to put in another excipient like a binder that sticks them together. stops them coming apart. Now, if you put too much binder in your formulation, then it still has to disintegrate in the stomach. So you need another excipient, maybe a disintegrant, which sucks up water. It expands 300 times the size, helps the tablet explode in the stomach. But then it may not flow well, so you have to add a glidant. And what happens, well, you definitely need a lubricant because you need the tablets to eject from the tablet press. And, well, if it's low solubility, you might want to add a surfactant. And then what about if it's unstable? And this is formulation development, okay? The formulation scientist is the Michelin star chef. He's the one that brings all of these materials together. And this is why I love formulation development. And there's nothing as rewarding as the achievement of getting a dosage form through development and onto the commercial market. So, where do we start? We've got these functional excipients that we may or may not need in our tablet formulation, and we need to make sure that they're stable. We need to show that there's some compatibility between the drug and the excipient. So what we typically do is take 2 or 3 diluents, 2 or 3 disintegrants, 2 or 3 surfactants, whatever, cover all of those things, and we do some excipient compatibility studies. And these can just be binary mixes of powders that we stress out with temperature, and then we'll do some HPLC analysis to see how if they're compatible or not. These are the most boring experiments that you will ever come across, and this is the most exciting result that I can find. These are just mixes of the drug plus another excipient. I'm not going to give any prizes for which one is bad. You can probably see it's discolored already. But we would do HPLC analysis on this and see how much drug is left after the excipient compatibility study finishes. This is the same experiment, but we've thrown a little bit of water into the mix as well. You can see that the effect is much more pronounced. And the reason that we do this is because our manufacturing process might be a pure dry process, or it could be a wet process, like wet granulation. So we want to see if moisture impacts our compatibility as well. These are the traditional ways of making tablets or capsules. The most simple way is direct compression, where you just blend all of the materials together. But remember that these things can demix. So it may be that we need a granulation process as well to get all of these materials to consolidate and stay uniform within the batch. We want to go from, where's my laser? We need a free-flowing powder to run on our tablet press. We need a free-flowing powder to run on our encapsulation machine. So if you have a material like this, we're going to have challenges with weight uniformity coming out of the tablet press. We want material like this. So if the drug is good enough and has good enough flow properties, and the question was asked earlier on, what particle size do you want? Well, I want a particle size and shape that means the drug will flow well, because this is the cheapest manufacturing process that we can come up with, and it is the fastest. If the particle size isn't good and we have poor flow properties, and that can come from shape of the API as well as particle size, we may not have any choice but to go down the path of a granulation process, wet or dry. So, just sticking with your theme of exploring early development, I just wanted to pick out some of the small-scale equipment that we use within formulation development. At the top, this is America. They typically have double cone blenders, and they have V-shaped blenders. You can fill those about 50% fill. They're not as efficient as perhaps the IBCs or tote blenders that we use in Europe. These would be these ones here. It's a bit like a cube with a pyramid on the bottom. And this one here is a small lab scale blender, which we have actually used for commercial manufacture. So going back to those niche indications, the orphan indications, we're actually starting to use lab scale equipment for some of our commercial manufacture now. This can take a 1 litre up to maybe a 15 litre IBC for just making small batches. Very, very effective. And even if you have to go down the route of granulation, at some point you have to do blending steps within your manufacturing process. So this will always come into play. These are the large ones from a commercial manufacturing organization. So they can go up to about 2,000 litres and would be used for some of the very large blockbuster medicines that we have had in the past. If we do go down the route of wet granulation, we can pick up small-scale high-shear mixers. Again, this one, you can attach a bowl of half a litre up to about 6 litres, and we can make small batches, hundreds of grams up to maybe a kilogram and a half. And in this high-shear mixer, we throw all of our drug and our excipients in together, and we wet it slightly with a granulating fluid, probably water, and generate a wet mass, which has to be dried before we can then fill it into the tablet press or encapsulation machine. So if you've worked in formulation development before, there's a reasonable chance you may have seen one of these pieces of equipment before. This is an aromatic Strea. So it's a fluid bed dryer. Hot air comes in at the bottom. You typically dry at about 60 degrees. Fluidize the bed of powder and just drive off the water. There is also the option to granulate in one go, so you can spray granulating fluid on top of the powder at the same time as drying it. It's a dynamic process, so that's a second way of doing wet granulation. Here's a more modern fluid bed. This will take up to about 5 kilograms. So you can still buy these today, and we were finding that we're using these not only for clinical trial supply manufacture, but they are creeping into commercial manufacture now just because of the batch sizes that are required. In dry granulation, there are many different suppliers. This is one of my favorites from Gerteis, a Swiss company. It would probably be the Rolls-Royce of roller compactors. They are very flexible, where the wet granulation is a batch process and the size of the bowl determines what scale of manufacture you're operating at. This is a continuous process, and you just keep feeding the machine and you can run as fast as you like. At the bottom end of the scale, you could even roller compact as small as about 20 grams of powder. On the other end of the scale, a mini pactor will, at maximum, churn out 100 kilograms per hour. So there's a wide range of production rates from this. I would say that we're starting to see less and less of wet granulation because some drugs are sensitive to moisture. It's an expensive process, lots of energy going into it. There's probably a lot more dry granulation happening nowadays. Then we have a tablet press, and if anyone has worked on these before, yes, this does look like a very small single punch tablet press. It's used in R&D, but it'll still produce 60 tablets a minute. It'll still produce 3,600 tablets an hour. You only have to run this for a couple of hours, and you've easily made enough stability supplies and clinical supplies for phase 1, phase 2 studies. We have even used this equipment for manufacturing commercial batches. Again, the example I gave before, it's only 3 batches a year, 5 kilograms for each batch. That's how small the market volumes were. As well as that, we have a rotary tablet press, and typically a lot of people scale up their manufacturing process onto a higher speed press. Some of the machines are very large. If you go to a company like Fete, which is one of the big tablet press manufacturers, they don't even make small rotary presses anymore. They're only concentrating on high volume production. But actually, you can still buy small rotary presses with only 10 stations that are suitable for small scale batches and even commercial batches as well. So, keep it simple. That's my advice all the time with formulation development. What are the basic goals for your formulation development of your solid oral dose form? So I'm just going to divide this up into the tests that you perform on the specification of your tablet, okay? The first is assay. You should be between 95 and 105% label claim in Europe, or 90 to 110 in the US. This is very easy to control. All you've got to do is make sure you weigh out the right amount of API into your batch. Straightforward. Just remember, we talked earlier about salts, we talked about solvents. You need to correct for that when you're dispensing, as well as you need a certificate of analysis for the API. No batch is ever 100.0% pure every single time. So if it is 97.8%, you need to correct for that as well when you're dispensing. The next thing is uniformity. And I've already talked about our multi-component formulation. So we just need an adequate mixing process for that. And we're obliged to take samples from the blend throughout and also from the tablet press to make sure all of the tablets are of the same strength coming off. Okay? Then we have degradation. And there's not so much we can do with this in that it is an inherent characteristic of the API. The stability of the API will determine the stability of the drug product. However, it's important that we only choose excipients that are compatible with the API. I explained that earlier. And that we choose the right packaging configuration. So you can choose something simple like a PVC blister, which is not really that protective, or you could go for aluminium foil blister pack, which is going to stop moisture getting into your tablet and causing degradation. So packaging is really the thing that you can work on with regards stability and degradation of your drug product. Appearance is another standard test. All you've got to do is make sure you have the right shape, size, and color of tablet. So you just need to choose the right punches, make sure you coat it with the right coating. You don't want the tablet to fall apart in the pack, so it's important that you make them hard enough and that you don't have any friability with your tablets, because you will get customer complaints if they start to try and pop them out the blisters and they start breaking apart. So those are the most basic goals of formulation development. The more complex ones are drug release, and then things like product line extensions and patient populations. Now, drug release actually doesn't have to be complex. If you are developing an immediate release dose form, and it's a good solubility drug, then what you're looking for is more than 85% dissolution in 15 minutes in your dissolution test. If you can achieve that, then that's absolutely fine. And that's about selecting the right amount of disintegrant in your tablet formulation. And if you've got a BCS class 1 drug, there's a fairly good chance that you're going to achieve that. The challenge comes when you have a low solubility drug and then you need some bioenhancement. And there's been a number of things discussed today: the use of salts, the use of hot melt extrusion, the amorphous form. That is a much more complex thing. And I'm going to give an example of how crazy and funky you can get with that afterwards. The last one is controlled release. And it probably comes more like a product line extension, where instead of taking 2 tablets a day or 3 tablets a day, the company might want to develop a controlled release dosage form so that they only have to give 1 unit instead. You're going to have to put a lot more drug into the unit form, 3 times as much if it was like 3 times a day. and then you have to formulate it in a different way that slows down the release of the tablet and slowly is absorbed from the stomach over the period of 24 hours. Then other complex goals, we may be asked by our marketing department to look at combination products, so maybe 2 drugs, and they may be compatible with each other, they may not. We have to work ways around that. And then we have alternate patient populations like pediatric and geriatric, where we have to develop different types of formulations for them. So I'm going to give a couple of case studies, if I'm still on track, bioenhancement and pediatric formulations. So bioenhancement, straightforward, use of surfactants. I talked earlier, amorphous formulations as well. Probably the most common is micronization, particle size reduction. And one of the things that I'm looking at the moment with a drug delivery company is a nano formulation, which is really quite interesting from a technical point of view. And in this, we are actually using physical vapor deposition. This is a technology, this is a technology that is used in the aerospace industry and the electronics industry. So under high vacuum, we evaporate the material and create a drug vapor. Yes, we're creating drug gas, okay? And then we mix up an inert carrier like lactose or mannitol, but we control the temperature of it. And we keep it at a cooler temperature, so when the drug vapor hits the carrier, it actually changes state from gas back to solid. And it does so as a nanoparticle. This is just a video that has been speeding up. But you can see the drug has actually changed to liquid, and now it's starting to vaporize off this under the vacuum chamber. We're operating at about 10 to the minus 5 torr, 10 to the minus 6 torr while we're doing this. And we're able to create nanoparticles around about 250 nanometers. And I know this is not a tablet, but you can make these powders and we can actually make them into tablets as well. But this is a project that we're working on at the moment, which is for Parkinson's disease. And sometimes Parkinson's patients develop off periods where their medication doesn't work. And the key medicine that's on the market today is an injection called Apokyn with apomorphine in it. And this is our first dog PK result. And you can see the red curve is the subcutaneous injection, okay? Now, the reason that we're developing this as a nasal dosage form, and it's a powder nasal dosage form as opposed to liquid, is that the drug can cross the blood-brain barrier much faster if it gets into the blood vessels in the nose. And we were quite astounded by these initial results because we're actually seeing faster absorption from a solid in the nasal cavity than you're getting from the subcutaneous injection. So we're making GMP batches next year, first quarter, and we have our first human studies planned in the first half of next year. So this is quite an exciting technology. It's quite funky. It's not going to be for everyone, but it just shows you how far we can push the boundaries when it comes to bioenhancement. Next one just is pediatric formulations, because everyone has to develop these now if they want to get their drug on the market. And historically, everyone goes down the route of liquid formulations, highly flavored, given with oral syringes and measuring spoons. What we've just done is we've piggybacked off our standard tablet compression and making mini tablets. These are only 2 millimeters in diameter. And you can put these into clinical studies with a simple counting stick, but also commercially, they are available in stick packs, or you can have counting devices. What we found was we've got probably 3 or 4 big pharma companies that now adopt this as their standard platform for pediatric medications, because they don't have to put lots of development time and resource into developing a solution or a suspension. They can piggyback off the work that they've already done for their adult dosage form, and then that saves a lot of time. They're made on a tablet press in exactly the same way, but you just have very, very expensive punches and dies. And they are very expensive, so they are. These things, there's 37 tips per punch. It probably cost about 6,000 euros just for one punch and one die. And then we can coat them as well if we need to. And this is the stick pack machine that we're filling all of our tablets in. So we actually have quite a number of commercial products on the market in this format. There are complexities with that. You have to have superior flow from your blend because it has to fill into the dyes at a very fast speed. And we may have to adjust certain things like lubrication because we have a very large surface area for very small mini tablets. But it can be done, and it's quite a successful way of formulating. I think I've, just looking at the time, my last slide I'll jump to. And the FDA have just put out the last year's results for the number of applications that have been approved. It's been quite a good year, a total of 54 new drug applications. And encouragingly, the vast majority of them are small molecules, 37. And over 60% of those are still oral dosage forms. So there's still a big market for tablets and capsules. And if anyone's working in formulation development, I encourage you to stay in this area. It's a very rewarding environment if you can get a drug to market. The other ones are the biologic license applications. Those are all biopharm molecules. So it could be monoclonal antibodies, gene therapy. Those are all injections. That'll not change. But there is still life in developing solid oral dose forms. Thank you very much.
A fantastic talk to finish the day. Questions here in the audience, please?
Thank you, Michael. Very nice talk. So you mentioned multiple times that it's still small molecule has still is important in the area, but we cannot ignore the fact that right now in the big pharma pipelines, like they spend more and more in biologic, and like they start new pipelines in biologic, and they have less and less budget in small molecule.
Yes.
So I just wonder, like, what's your feeling and what's your opinion about this? And yeah.
From what I know in the biopharm, it is very easy to destroy a batch. Interestingly, we were talking about tox and PPE. In that manufacturing situation, the batch of material is more of danger from the operator than the operator is up for the material. It's the opposite way around for small molecules. And because they're very sensitive to manufacture, they have to throw huge amounts of money at that. So if you're measuring it in terms of cash, biologics, they're worth so much money. So you're not going to not work on that area. You have to, if you can. But if you have an indication and you can make a tablet and you can make an injection, and I offer both of them to you, which one do you want to take? If someone says you will die if you don't take this injection, you'll take the injection. Yeah, of course. And if it's so life-changing that one injection solves everything for the rest of your life, yes, you'll take the injection. If you have a headache, no, you're going to take the tablet or the capsule. And I just say that, I mean, consider your family, maybe your parents or your grandparents. I think you'll still see that they are still counting out their tablets and their capsules at the breakfast table in the morning. So I suppose I'm just trying to say is I don't think that the tablets will stop. I don't think the small molecules will stop.
Thank you.
Any other questions?
I have a curiosity about the very small blenders that you're using for commercial production. How do you verify uniformity of blend with such a small batch size? Do you use PAT or special analytical techniques?
You can use PAT, but quite often we'll just take samples. If the blender gets so small, you may only be able to take N of 3 instead of N of 6. But your typical sampling plan for a blender would be 6 samples, spaced at the top and at the middle and one at the very bottom. You just may have to change that if you get to a batch. It's like a 1 liter IBC. But the density of most powders, going to be, let's just, for simplicity, 0.5. You know, even if you're making a sort of a larger scale liter phase clinical batch at, you know, say 2.5 kilograms, that's going to be a 5 liter volume. You're probably going to need about a 7.5 liter IBC. So you still have space to take, easily to take 6 samples when you get to that size.
Thank you. Any other questions?
So I'm particularly interested in your mum counting the tablets for your dad.
Yep.
You know, because I know situations where there are quite a few tablets to count, and the people who are supposed to take them can't really manage it. So I can well see the injection taking the place of these tablets because it's a nuisance. And they're much more complicated to make, the bioavailability, the absorption of the small intestines. There's so much complexity there for a tablet to work, where an injection is straight into the bloodstream, and it's working within seconds.
True, and it's an invasive technique. There was a newspaper article just a while back there. There's a nurse that was taken to court, very unfortunate, and she didn't wipe the patient's skin with the alcohol, and then with the injection she got sepsis. Now, thankfully the patient didn't pass away, but she was hauled in front of whatever body it was, you know, checking did she follow procedure and everything. That was in a care setting or a hospital setting. So I wouldn't say that injections are without risk.
But then you could forget to take the tablet, couldn't you?
You can forget to take the tablet, and you can forget to take the injection. Well, actually, I was referring to my mother-in-law. My mother actually does have Parkinson's, and she's in a home. And such is the challenge for her, the care staff actually have to give her a patch, so that because there's times when she'll fall asleep, and you literally cannot give her the tablet. So I admit there's circumstances like that. So they give her a patch of Matapar, partly so they can prove that they administered the medication and they don't get in trouble. But yeah, I think you can make arguments either way. You know, I don't think that injections are just the answer to everything.
Okay, one final question before we close for the day. Michael, when you put the slide with the formulation or everything that goes in the tablet, I think I counted 10 or 12 different components there. Obviously, you know, we typically work on some early preclinical formulation. They are a lot simpler than that. What would be typically your kind of composition for a preclinical vehicle?
for
a preclinical vehicle that we're going to be using, maybe surfactants, maybe we're going to use a polymer for amorphous dispersion, but not as complex. What would be your recipe if you went for preclinical?
In my experience, a lot of preclinical studies are conducted on rodents, so they're often given with just suspensions, and it may be as simple as water and a suspending agent like carboxymethyl cellulose, and it doesn't have to be any more complicated than that. You're not going to be able to dose tablets easily to a rodent. They're just too big. Well, you probably do the mini tablets, but you get to that level of complexity when you have to, and that, for me, comes after the preclinical stage. That is going into your clinical studies.
Obviously, I was thinking from the person who is developing the formulation, not certainly the one administering to the animals, but thank you very much. I think we're going to leave it here. I think an opportunity to thank Michael again and all the speakers.